In this presentation, I'm going to talk about the work package 3, which is about membrane flexibility. In this work package, we are working to track regulation phenomena at the level of the organism in vivo in a concerted manner. For this, we are using different microscopic and spectroscopic methods. The results obtained from this work package will be combined with those from work package 1 and work package 2 for the proper assessment of spectroscopic signals in many cases. At the same time, the outcome from all work packages will be used to develop an efficient monitoring system for the productivity of microalgae. We are in total 4 researchers working in this work package. I will discuss about our individual projects sequentially. So the first project is about photosynthetic membranes from molecular to membrane organization. The objective of this project is to have precise information on the conformation and interaction of the photosynthetic pigments in intact plants and algae. In this regard, the researcher is using resonance Raman spectroscopy, fluorescence line narrowing, and an original method for super resolution microscopy reaching about 30 nanometer resolution on naturally fluorescing samples. I am working on the second one, where I focus on investigating the subcellular localization of photosystem 2 biogenesis and repair processes in cyanobacteria. The biogenesis of photosystem 2 is a complex multi-step process. It requires precise assembly of all the protein subunits and pigments. Due to the high sensitivity to light, photosystem 2 also undergoes continuous damage and requires repair cycle to restore its photosynthetic capability. Although photosystem 2 is one of the mostly studied photosynthetic complexes, the subcellular localization of its assembly events is not clearly understood yet. However, to have a better understanding of the overall thylakoid membrane architecture, it is important to know the sites of photosystem 2 assembly. So to acquire high-resolution information, I aim for probing different assembly factors such as CTPA and FTSH. The role of these two proteins in photosystem 2 assembly events are shown in the figure. My original idea is to tag them with fluorescent proteins for the visual identification through different microscopy approaches like confocal microscopy, structured illumination microscopy, and atomic force microscopy. Our third project is about changes at the membrane level during NPQ and state transitions. In cyanobacteria, light harvesting takes place in the core antenna complexes in addition to the membrane external phycobilism antenna as shown in figure 1. Differences in the absorption spectrum of these two antenna systems and their relative quality of each photosystem disturb the balance of the excitation between two photosystems under changing quality of light. Under such changing light conditions, cyanobacteria dramatically restore the balance of excitation between the two photosystems through a process commonly referred as state transition. Picosecond time resolved fluorescence studies are performed on bulk of cells to discern the differences between the states in terms of excitation energy transfers. Mobility of phycobilism antenna and photosystems are shown in figure 2 is also investigated. Individual cells are studied with fluorescence lifetime imaging microscopy in order to investigate if there are structural changes in thylakoid membrane accompanying the state transitions. Lifetime imaging also provides the information about the distribution of photosystem complexes in the thylakoid membranes. The last project is about the involvement of secondary metabolites, where we are trying to address the question of how the culture parameters affect the production of a potent secondary metabolite in nostoc. This metabolite is cryptophycin, and we can see here on the figures its main analog, cryptophycin 1. This compound was discovered by Schwartz 30 years ago, and for the first time it reached for the clinical trials in 2002. However, it fails due to toxicity issues. Its anti-cancer potential activity is based on the fact that it stops the polymerization of tubulin and it causes apoptosis. Its total chemical synthesis was multi-step and it faces purification issues, so it is not applicable in the industrial scale. In that way, we are trying to isolate the compound naturally. We use the research on photosynthesis as a tool to help us study the cultural condition of the microorganism. So then we stress the conditions like light intensity, etc. to draw the related curves and finally overproduce the metabolite.